Chetelat. I'm director of the Tomato Genetics Resource Center. So the Tomato Genetics Resource Center is affiliated with the National Plant Germplasm System, which is part of the USDA. And we have about 4,500 accessions in our collection. And we distribute seed around the world to researchers and plant breeders and educators working on tomato. Each year we receive about 250 seed requests, and we send out roughly 5,000 to 7,000 seed samples. So the collection is quite actively used relative to its size. We're standing in our greenhouses here at UC Davis. This is the greenhouse where we do seed regeneration on our collection. The material that we have growing here is mostly wild species, wild tomato relatives right now. We also have a lot of mutants in the collection and other types of genetic stocks. The wild species are used for all types of traits that can't be found in cultivars and varieties of cultivated tomato, primarily disease resistance traits. And I'll give you a few examples of that. So we're standing in this garden of wild species in our greenhouse, and one of the wild relatives that we maintain is called Solanum pennellii, and it's native to Peru. And this species has been extremely useful for plant breeding, for breeding new varieties with disease resistance and fruit quality traits that are important to either to growers or to consumers. So this is an example of the species. One of the accessions that we have of this species has been the source of resistance to a race of Fusarium wilt. It grows in the soil and it infests tomato roots and causes plants to collapse. Things like heirlooms and other older vintage cultivars were not resistant to Fusarium wilt. Beginning in the 1940s, breeders transferred the first resistance to Fusarium from one of the wild species, not this one. And that was the first Fusarium wilt resistance trait, Fusarium wilt race 1. And then that was overcome by the pathogen. That resistance gene broke down, and breeders transferred into tomato a second resistance gene, Fusarium resistance to race 2. Eventually that broke down, and then there was race 3 of Fusarium that became a problem. And this species was identified by researchers in Florida as a source of resistance to Fusarium wilt race 3. And it's donated this resistance gene over a lot of years of breeding and selection that confers resistance to race 3. And this trait now has been transferred into a lot of the processing tomato varieties that are grown commercially in California. And I think it's fair to say that without that resistance gene, you couldn't grow processing tomatoes in many parts of the state today. This species, Solanum pennellii, is, besides being a source of disease resistance, has also been a source of improved fruit quality. Specifically, breeders in Israel found that it contains a gene that elevates the fruit sugar content. And this is important to growers of processing tomatoes because they're paid not only for the yield, but they get a benefit for the soluble sugar content in the fruit. And what happens when the fruit are harvested in our production system here in California, they're trucked to the processing plant where a lot of the water is cooked off of the fruit to concentrate it down to tomato paste. So the less water they have to evaporate to concentrate to the standard tomato paste, then there's an energy savings involved. And this wild species, Solanum pennellii, contains a gene that increases soluble solids content roughly by 20%, which is really major improvement without reducing yield. So normally, oftentimes when you increase the fruit sugar content or other aspects of the carbohydrate content, there's a trade-off with yield. So the more sugar you have in the fruit, then the less fruit the plant can produce. But in this case, there's no penalty to yield. So it's really been kind of a miracle trait from this Solanum pennellii wild species. And ironically, the fruit that it produces are small green fruit that really have an unpleasant taste and texture. They're not really considered edible, but they contain, it contains this gene that's useful for improving the fruit flavor and quality of cultivated tomato. This is another of the wild tomato relatives. It's called Solanum chilense, and it grows in Chile, as the name suggests, as well as into southern Peru. And this has been a useful wild relative for breeding for disease resistance. It donated a gene for resistance to powdery mildew, which is a fungus that affects the leaves of tomatoes that are grown here in California and reduces the yield, particularly in fields of processing tomatoes. Beginning in the 1970s, researchers in Bulgaria made the first crosses with this species onto cultivated tomato, and over a period of a couple of decades transferred various traits, and including this resistance to powdery mildew. And now this resistance trait has been bred into a lot of the commercial cultivars that are grown in California, and it helps maintain the yield in these fields that are affected by the powdery mildew fungus. And of course, the alternative to having this disease resistance trait is spraying fungicides on the leaves of the plants. So by having this resistance in commercial cultivars, not only is yield higher or more stable and maintained, but also you have the need to apply fewer pesticides. This is Solanum pimpinellifolium, also known as a currant tomato. It's the wild ancestor of cultivated tomato, and it has these very small, kind of pea-sized fruit. You can see they're red, just like a regular tomato, just like a small version of cherry tomato. The great thing about this species is it crosses freely with cultivated tomato. Very easy to make hybrids and to breed traits into cultivated tomato from this closely related wild species. And it's been the source of the first disease resistance genes that were bred into tomato, starting at the very beginning of kind of modern tomato breeding in mid-20th century. Specifically, the first resistance to Fusarium wilt and the first resistance to Verticillium wilt. Those are two vascular wilts that affect tomato in many parts of the world. Since then, it continues to be used for other traits, including resistance to salt tolerance and some other drought tolerance, but those have not made their way yet into commercial cultivars. This is another of the wild tomato species, Solanum peruvianum, and it's native to Peru, as the name suggests. And it has these small green fruit with purple stripes. It's been a really important species for tomato breeding. Several of the first disease resistance traits came out of this species. One of the most important ones is the nematode resistance gene. This gene is called MI, and it's the only source of resistance to nematodes in commercial cultivars. When you have nematodes in the soil, as I do in my backyard garden here in Davis, you really can't grow varieties that don't have resistance. Basically, the nematodes, which are microscopic worms, infect the roots, and you get these galls on the root system, and the plant yields very little and eventually collapses. This resistance gene was bred into cultivated tomato from Peru, Solanum peruvianum, in the 1970s, and it's still widely used. Many of the cultivars grown in California have this resistance trait. Another trait that came out of this species is resistance to tobacco mosaic virus, or tomato mosaic virus, TMV. And this is one of the two main genes for TMV resistance, called the TM2 gene. And again, that gene has been around for a long time and is still one of the main sources of resistance in commercial varieties, particularly in protected culture, in greenhouses, where plants are contacted a lot, they're handled a lot, and so they're susceptible to being infected with these viruses that are mechanically transmitted. It's also been a source of resistance to tomato spotted wilt virus. That's the virus that we have here in California. It's spread by these small insects called thrips. And this species, Solanum peruvianum, was the source of the SW5 gene for resistance to tomato spotted wilt virus, and still the only source of resistance to that virus that's in commercial cultivars. This is another wild tomato species. This one is called Solanum cheesmaniae, and it's native to the Galapagos Islands in Ecuador. It only is found on the Galapagos Islands. And it has these small orange, yellowish fruit about the size of a small cherry tomato. And one of the unique traits that's been transferred into cultivated tomato from this species is related to the separation of the fruit from the plant. So this species has a mutation that abolishes the normal pedicel joint. That's where the fruit would fall off the plant. and you would have that little piece of calyx with a bit of the stem of the flower cluster attached to the fruit normally. Well, this species doesn't drop its fruit, so that pedicel joint is missing. And that's useful for machine harvested processing tomatoes, because when those are picked off the plant by the machines, they're shaken off. You don't want that little piece of stem on the tips of the fruits, because those would puncture and wound other fruit when they get loaded into the truck bins and shipped to the processing plant. And this gene, it's called the J2 or jointless 2 gene, was first identified by Charlie Rick in the 1950s and bred into cultivated tomato. And then that gene was picked up and introduced into processing varieties. And now a lot of the processing varieties in California that are grown have this mutation, this trait that abolishes the pedicel joint.